Welcome everybody. This is we are now over a year and a half into doing our Chestnut chats, and I really appreciate you all for joining us. I see a couple of folks who are continuing their perfect attendance streak. Russell and Carl, thanks for coming, as you guys do every month. It's great to see you all on here, and a lot of other really familiar names. I wish I could shout out to everybody. But today we're going to welcome Chuck Ray from Penn State. He is a wood science professor of wood science. Well, it used to be professor of wood science. What's your title these days, Chuck?
Yeah, I'm associate professor of used to be wood operations research. I guess I say wood science nowadays. Nobody ever tells me anything around here, so I just go by what you want to call me.
Well, we'll say associate professor of wood science, and he's going to talk to us about chestnut wood ID and what he's been doing. So this is a shameless plug for me, for Sarah. I'm going to share with you guys this chestnut board. So this is what got me involved in American chestnut. It's a wormy chestnut board that my pawpaw salvaged from a one-room schoolhouse in southern West Virginia where I grew up. And I'm again sporting my West Virginia earrings. But he went around, and whenever they tore down old one-room schoolhouses, he would salvage all the wood, but particularly the chestnut out of it. And so I've still got a little stash of this stuff,
That's
100-year-old wormy chestnut. So I'm excited about this. To welcome Chuck. So a little bit of housekeeping, and then I'll turn it over to Lisa for a proper introduction. So as always, we are in a webinar format. So we can't hear you, we can't see you. If you're not Chuck, Lisa, Sharon, Kendra, or Sarah, we can't see or hear you. If you have a question, please put that in the Q&A. If you just want to chat, make a snarky comment, make positive comments, talk amongst yourselves, you may do that in the chat. But if you have a question, do put that in the Q&A. That's how I'm going to keep everything organized. I tend to hold all the questions until the end. I do keep an eye on it though, so if there's something that's really confusing, I'll interrupt Chuck and throw out the question. But typically I hold them all until the end. And there was a previous question about how long these go. Usually we have about 45 minutes for a talk, and then we chat for 30 to 45 minutes more. So typically we go a whole hour and a half, but we sometimes end sooner depending on how many questions you all have. Mahali, I hope that's how you say your name. No, we cannot see you. We can't hear you. You guys can put on NPR in the background, have your lunch, whatever you typically do.
That's Mihai down in South Carolina. Now that's a real wood scientist there. So welcome Mihai.
Nice.
He's the editor of the journal World of Wood. He's a great guy.
Fantastic. Well, thanks for coming and joining the Chestnut crew. Well, with that, I think that's all the housekeeping we need. If you have any questions or anything about technical stuff, throw that in the chat. If you have a question for Chuck, throw that in Q&A. Other than that, I want to turn it over to Lisa to introduce our topic, and then Chuck will have you take it away.
Thanks, Sarah. Welcome, Chuck. We're so delighted you're here. A lot of interest in this. I had, look at that, 178, including the panelists. That's a pretty darn good turnout. So we're excited to have you here. It's really, one of the things I've noticed about the Chestnut family is the number of artists that we have, and having gone to art school, I keep an eye on them, and we have some fantastic woodworkers out there. So I'm sure several of them are on this call, and thank you for joining us, everyone. Chestnut Chat has been an enduring new feature of COVID. Sometimes there's positive things out of COVID, not very many, but Chestnut Chat's one of them. And I again want to thank Sarah, Kendra for helping us manage the questions, Sharon and Jules for the support that they give this wonderful talk, and hope you all are having a good fall. And with that, I'll turn it over to Chuck. Take it away.
Thank you, Lisa. It is crazy to me. I'm looking at your numbers. I can't believe that 184 people now are on the chat here. Mihai, I think you can relate to this. That's about 10 times the largest audience I've ever had for a wood science talk. And so maybe hopefully I won't get stage fright here, but I'll try to pull this up. If you guys remember the old movie Smokey and the Bandit, there was a great song there, and the guy said, we have a long way to go in a short time to get there. And that's what we've got here. A lot of slides to get to the final conclusion, and so I'm going to have to kind of go through them pretty quickly. And hopefully we'll get to where we can answer questions. Is it showing up correctly now, Sarah?
No, I am not seeing your screen.
They're not. Okay. Let me go back. Where am I here? Share
Now I'm going to have
screen.
that song in my head.
Sorry.
That's all right.
Great movie, great classic movie. But yeah, but you're not old enough to know about that movie, Sarah. Come on.
But I grew up in, my father loved it. We watched it constantly.
Let's see. Am I supposed to click on that? Is that share? Oh, okay, I see.
There it is.
Okay, good.
Great job.
How about if I click that? Are we looking correct here?
You're looking great. Thanks, Chuck.
All right. Well, welcome everybody. We here are on the American Chestnut Foundation Chestnut Chat. Thank you for joining me here today. And we're going to talk about wood quality and hybrid chestnuts. Everybody, I assume everybody for the foundation is familiar and probably active with the hybridization effort to bring American chestnuts back to the North American forest there. And I saw these two pictures and I thought about it in terms of the wood issue that we're going to discuss today. You know, when you go and find some old barn wood, it's chestnut wood, it probably came from trees like this here, right? And most of the specimens that we look at nowadays from the trees that we're growing are coming from trees like this at best, right? And the question that it brings to mind is, well, is the wood from this, or is the wood that we're growing in here really able to tell us how it's going to turn out when the trees get to be like this, which I hopefully they will, thanks to your efforts. So that's what we're going to try to discuss and examine and discuss here today. The other thing that this picture made me think of, and I think for me, it put something in perspective that I had never thought about with respect to your work at the American Chestnut Foundation. And that was that when pictures like this were taken when the chestnut forest was here in the eastern part of North America, there weren't many people that were going around taking pictures of it, right? There were a few pictures like this, but generally speaking, most people, unless they lived in the East and went out in the woods, they never really saw this kind of thing. And so when 100 years or so ago, they started dying off, maybe it didn't quite have the impact. I thought about what would happen today. I mean, people today, everybody knows about the redwoods out on the West Coast, and because of YouTube and you know, millions of photographs that we see every day, everybody is familiar with the redwood forest and how significant that is. Just imagine if there were, and I'm going to knock on my desk when I say this, just imagine if there were some kind of a redwood blight that went through these days and wiped out the redwood forest. Think about how horrendous that event would be perceived, and how many millions and millions and millions of dollars would go into efforts to try to stop and turn that around. And yet, essentially, the same thing happened here on the East Coast 100 years ago. And so the efforts that you guys are going through, which are somewhat in relative obscurity compared to all kinds of other things that are frequently promoted, probably was way more significant than we ever give it credit for. And it certainly is so when you examine the skeletons of a lot of the old wooden buildings and you see how much chestnut there was actually used back in those days. So just a kind of a neat thing. Welcome to the chat here. There are some things that we need to be clear. We need to talk about some terminology here. Now, I certainly don't want to insult everybody. I'm sure there are a lot of woodworkers and people that are familiar with wood terminology, but I'm going to assume that some portion of you guys are not familiar with some things. And so we're going to have to talk about some terminology to clear it up so that the results we're going to see today make some sense. The first one is hardwood and softwood, difference between them. The next dichotomy is heartwood and sapwood. We have early wood and late wood, which is sometimes called spring wood and summer wood, but it's probably more correct to call it early wood, late wood. We have juvenile wood and mature wood. We have green wood and dry wood. And so if you're at this point and you're going, oh, well, I didn't know that there was a difference. Like, for instance, I always thought early wood and sapwood was the same thing, or I always thought heartwood and mature wood is the same thing. Well, if now you're questioning yourself, you should be, and that's what we're going to cover here in the next few slides, because those importances are real and very important in terms of how we evaluate what's going on with our hybrid chestnuts here. We'll start out with hardwood and softwood, which is just sort of a general thing that I get questioned. This is the one I get asked about the most often is, what's the difference between hardwood and softwood? How can you tell? And And so very generally, we're talking about the difference between angiosperms, which are deciduous trees, and gymnosperms, which are coniferous trees. And that's really the primary and only real difference between hardwoods and softwoods. There's no real relation to the hardness or softness of the wood at all. And I do have a blog post. If you want to go look at my blog, it's gowood.blogspot.com, and you just go up in the little thing there and search for hardwood, softwood, and it'll pull up a pretty long blog post describing the biological differences between them and also how the terminology developed over time there. But here you see a nice little slide here of a couple hardwoods, oak and mahogany versus spruce. You know, sometimes you can see that looks like sometimes I think that they look inversely on the colors, but they're actually the same thing with light early wood and darker late wood, and we'll talk about that in the slide to come here. We have a ringed, diffuse porous hardwood here. The trees down the tropics tend to not have distinct rings because they have basically their growing season is all year long there. And so you have what's developing a diffuse porous scenario with no growth rings here. So that's a pretty neat distinction between the trees. This is a softwood here, bald cypress, which I've done some work on in my lab here at Penn State on. And it looks very much like other softwoods. The thing I did in my lab here was an interesting project. We had to got involved in a study where you may be familiar with the product bark mulch, and a lot of the bark mulch is pine. Some of the companies started marketing a cypress bark mulch product a few years ago, and according to the agencies that monitor product truth in marketing and things like that, the cypress mulch had to have at least 51% cypress in it, and yet some of the companies were questioning what one particular company was adhering to that 51% or not. And so I was able to gather a bunch of bags of the product from around the country, screen them out according to size, look and determine how much of it was actually cypress versus pine. And it was relatively easy, even though these are tiny pieces of bark mulch. Let me see if I can get my cursor again here. Yeah. Even though these are very small pieces, so you can see this is only 2 millimeters, basically like the whole piece here might be only 4 millimeters wide. As long as I was able to get a clear profile of the cross section, or we call the transverse view of the wood, then I was able to determine whether it was cypress or pine, basically because the pine had some resin canals all here in the early wood, and they were pretty distinct, nice large pores that were filled with resin. And since I didn't see those here, I know that this is a bald cypress piece of mulch. And so then I was able to break them out, differentiate them, weigh them, and determine what percentage of the mix then was cypress versus pine. And then we have an interesting pair of slides here, pictures from I just took the other day from downstairs. We have a large podium that was made from a piece of sumac that was harvested about 100 years ago. And these are two pictures that were taken just by my phone that show just about the same view. I think this one's a little bit closer, but this one here on the right is a picture in the natural daylight that's in the atrium of our building. And this one on the left was when I turned the flash on to my camera. And you can see just the change of the light from my camera. You can see how much the color of that wood changed. And so that was one of the issues in our study, is what is the real color of our chestnut hybrids versus the Chinese chestnut and the American chestnut. And we're going to talk a lot about that as well. Okay, so now we'll talk about heartwood and sapwood. Heartwood is the general portion, the central portion of the stem that's no longer conducting water, serving food to the tree. And sometimes it's called the dead wood in the tree. I don't like to call it that, but some people do. And then the sapwood is the outer living portion of the stem where water conduction and food storage occurs. And the relative size of heartwood and sapwood varies according to the growing conditions. So here I took a picture, a terrible picture, but they put a piece of plastic up in front of our cross section here. This is about a 3-foot across piece of hemlock here, and that was harvested here in Pennsylvania. And what we can see here is a big central core of heartwood, and it's really nice wood for the woodworkers and things like that. but it is dark and very distinct from the outer line of sapwood, where all this, where all the fluid and carbohydrate movement is occurring. And that's distinct from what we see up here on hardwoods more commonly. But this could also look like this in softwoods. There's no real distinction there. I'm just showing the two different kinds of how heartwood and sapwood present themselves. This is a very distinct line. This is much more gradual in this sumac podium. You can see the heartwood here, but you also see like another band heartwood developing out here, sort of transitioning gradually. Even though this sumac specimen is over 100 years old, you can see that we still have a gradual transition to heartwood. And if the tree had kept growing another 100 years, this would have all been solid heartwood as well, and you'd have had more sapwood out here on the outside. So that's heartwood versus sapwood. Now we're going to talk about early wood and late wood, which is completely different. Early wood is this lighter wood here that's formed from the time the sap begins to rise in the late winter until roughly the summer solstice, when the days begin to shorten. And the new cell generation responds to this new cell generation, which is in chestnut, these rows of rings here, we call big pores. The new cell generation responds to lengthening days and plentiful water in the spring. Oh, turn my phone off here. And turn that off. Thank you. There we go. So we call these the first cells produced every year, the pore. And chestnut, like oaks and black locust and other kinds of hardwood, other species of hardwood trees, are what we call ring pores. These pores develop clearly defined rings, as opposed to semi-ring porous and diffuse porous trees, which fall in a different category here. But these are chestnut, and we see the difference between these. This is one growth ring as opposed to down here. This is another growth ring of a different specimen, and we can see the difference where we have a clear delineation of early wood versus late wood, wood that's grown in the early part of the growing season versus in the late part of the growing season. And again here, early wood, late wood, and as defined by one ring, clear ring of pores that delineates 1 year from the next there. Okay. So that's early wood and late wood. And late wood is formed during the later part of the growing cycle when less sap flow is occurring. The days are shortening. The tree growth is primarily in the form of these late wood vessels, which are late wood cells that are thick, developing thicker cell walls. And so what happens there is actually when we saw or sand the piece of wood, these thicker cell walls get burnished more from the process and darken up more than these early wood cell walls, which are much thinner cell walls there. And this transition between early wood and late wood can be either distinct or gradual, like we see here. Then we have juvenile and mature wood, which is something that most people probably don't know about it unless they're very familiar with wood. Juvenile wood is the wood that's grown, that is developed during the early part of the tree's life, when it's really growing as a juvenile, as a young person, for instance, and it's got a lot of sap flow. It's growing much more as a relative to its diameter than older trees do. So if we're looking at this piece of juvenile chestnut here, we have a higher growth rate with wider rings that are usual to trees in the first 8 to 12 years of life, versus this is a piece of mature ash here. And we see a lower growth rate, narrower rings, usual to mature trees. Okay, you have to remember that the tree itself, because these rings are so much bigger than these rings, there's still a lot of wood, maybe possibly as much wood being added in the mature years. It's just the ring spread out over a much wider ring than it is here. In the juvenile wood, we have more variability in the growth ring shape versus something that's more concentric and uniform in mature wood. we have greater microfibril angle, which is the fiber bundles. If you look at them here in the transverse view, they tend to be going out at different directions in the juvenile wood, as opposed to when they get over here into the mature wood, they tend to be more uniform, pointing in the same direction there. The juvenile wood is lighter in color due to a higher percentage of early wood. The early wood, again, tends to be lighter. And so because in the growth ring, because the growth ring is wide with a lot of early wood, it tends to be lighter in color. Whereas here in the mature wood, the early wood and late wood is about 50-50. And so you get a darker color coming out of that. And typically, in most of the wood science textbooks, they say that the juvenile wood is lower density and the mature wood is a higher density. And chestnut is an interesting exception to that. We'll talk about that more in future slides here. This picture was really interesting. The specimen was supplied to me by Gary Carver, and I'm going to talk more about Gary in a minute. He observed something really interesting. He said, no, 25 years before the release. So we have 25 years of growth rings here. So the tree was basically laying kind of not grown at all in an understory. And then the canopy was cleared and the tree took off. And it does show, it's interesting that once the tree starts taking off, even though the tree now is 26 years old, it's still in its growth mode. It's still developing rings that have the same properties, are essentially juvenile wood. And so that's something that I had really never thought about until I saw this specimen. It can happen that way, but generally these juvenile wood portions of the tree are the first 8 to 12 years or so of the growth of the tree. Okay.
Hey, Chuck, before you leave that, we had a question about what is a microfibril angle?
Well, it's just if you think about the trees, the cells in the tree, which are growing in fiber bundles and things like that, these are growing, and because of the different kinds of environmental impacts on the tree as it's new, the angles of the fibers themselves are more impacted by outside influences like cankers and insects and things like that. So you tend to see more variability in the angles of the fiber, versus once the tree is mature and it's growing kind of generally the same way every year, then those things, the angle itself tends to be more uniform there. Okay. So, and also we have a distinction between green wood and dry wood, because one of the issues in the study was, boy, this Chinese wood is sure green, and why is that? So green wood, and that's confused in terms of when you buy firewood or something, you're buying green wood versus dry wood. Green wood typically has been harvested in the last year or so, and it has a moisture content higher than 20%, and it's going through a natural process of drying down to equilibrium moisture content, which is from 10 to 16% in different parts of North America. Most of North America, it's between like 12 to 14%. Here in Pennsylvania, around 12 to 13%. And the green wood is so called because many species exhibit a greenish hue when they're at this higher moisture content. And so Chinese chestnut, for example, appears very green when it's freshly cut, as does sycamore, striped maple, yellow poplar. Okay, very greenish woods when they're freshly cut. Dry wood then is wood that's been aged or seasoned or dried in a kiln, and now it's below 20% moisture content, and it's nearing the equilibrium moisture content. And natural air drying can be sped up by minimizing the moisture exposure in a drying shed, an industrial pre-dryer, or climate-controlled room. And dried wood typically changes color from slightly to a lot, depending on the species and the cut of the wood. We had a fortunate thing happen on this particular study because we were delayed a year because of COVID. Then all of our specimens were in my lab at a controlled climate for over a year, and we got everything to EMC basically. And so that was helpful because some of the specimens from the orchards were freshly harvested, and they were pretty green. And by the time they went through a year and all the bugs emerged from them and things like that, they had a very mature or a dried appearance to them. Okay. I threw these next two pictures in just because they were neat pictures that came from offline, and they have some more terminology in here. Again, we'll see the Heartwood. We see the Heartwood here, which is the darker hue, versus the Sapwood, still living, still conducting moisture in food. We have the pith, which is the first year of growth, which is the central ring of the tree. And we have three terms here that we're going to talk about a lot in the rest of the presentation. That is the transverse view, or the cross section of the specimen, the radial view, which shows the same relationship of growth rings, but it's from the side instead of from the top. And then the tangential view, which is sort of a flat, if you think about lumber, it's flat sawn lumber, where you see these wide growth rings. And typically on the tangential view, some of those that I had in the study, I couldn't use them in my growth ring measurements because the growth rings were too wide under the microscope to capture more than one ring or one complete ring. So yeah, three different views here that have kind of three different properties perhaps, but I think I'm going to show you that we can still collect the same data off of all three views. And this one I included because, let me back up. By the way, this was interesting because in this particular picture, we can see the Heartwood is this portion, the Sapwood is this portion, but I would call from here back all juvenile wood, because you can see that these rings here kind of had the same general properties, width and things, ratio of early wood to late wood. But once you get out here, now you're sort of more into the mature wood portion of the tree here. And probably if you count these, you're probably getting to 10 or 12 somewhere right out around here. But it does show clearly that Heartwood and juvenile wood are not the same thing. In a lot of trees, like I said, the Heartwood could be all the way here and encompass a lot of mature wood, as well as all the juvenile wood. Okay, so that's interesting. This one has the addition of rays in it. These rays are going out, and rays are an interesting characteristic to us because they are sort of a distinguishing characteristic between chestnut and the oaks. A lot of times in the lab, people will send me stuff from a barn here and they'll say, is this chestnut or oak? And of course, once you know, it's really pretty easy to tell the difference. But if you don't know about rays and their how they appear, then you don't know the difference there. But you can see how rays typically appear on a transverse view here. You see these, and that's typically what you'll see in pieces of oak. I'm looking at my desk here and I see these nice wide rays, which give it that oak look to it. This is looks like a piece of softwood. I'm going by the bark, but you can't tell really. It's just a general drawing. But that's how the rays pop out here. Okay, they grow out like this, and it's important because I have a nice slide later that I'll show you that shows you that the rays are coming out from the pith in a radial direction. They're radiating out from the pith. So we'll see that again here, and I'll show you the difference between oak and chestnut when we get to a good slide here.
Hey Chuck, I got some simple questions here for you,
Sure.
if you don't mind. So what
Sure.
causes heartwood to form?
I'm not sure I understand the question. What is heartwood to form?
What causes it to form?
Oh,
So why does a tree go from juvenile wood to heartwood?
yeah. Well, that's an interesting question, and where there's a genetic component to that, and there's also a growth component to it. It's really a function of how much the tree requires in terms of fluid and food movement up and down the stem. One of the things that you'll see, I think, typically is that in a plantation, for instance, where the trees are getting a lot of light, they're getting a nice, especially when there's a lot of water available to them, rainfall and things like that. You're generating a larger crown, a lot of leaves, and you're getting a lot of transpiration of the leaflets and a lot of fluid uptake through them. So you have a lot of sapwood staying active, and so those cells that are transporting everything up and down stay open and flowing in order to feed that tree, the crown. in trees that are growing in the forest, they, as a percentage, if they're growing with smaller crowns or more bunched up, they don't need as much fluid transportation relative to the size of the stem as the more open grown trees do. And so what they'll start doing is those inner layers of wood all of a sudden aren't sucking up a lot of wood, and they start clogging up and collecting. Basically what happens is they just get poured up because it's no longer flowing, just like an old piece of plumbing or something like that. If you don't have good flow, then things are stopping up, and you start picking up resins and gums and things like that would plug up, and then there's no longer any flow in there. But as long as there's a large requirement of a big crown that's keeping things flowing well, then you're going to have a larger proportional amount of sapwood in the tree to the heartwood than you would in maybe a forest grown tree there. Okay.
So two more questions. Does the tree make any effort to keep heartwood alive, or is it truly dead?
Does a tree make any effort to keep it alive? Well, as I said, stuff is flowing up and down as long as it needs to flow to feed the tree. And when the tree no longer needs it, as appointed by the amount of branching and leaves that are on the tree, then it goes ahead and quits using those cells in the middle. It also has to do, as the crown grows out, your root structure is also going to grow out then, and which means that underneath and right in the middle of the tree, you're not going to have a lot of fluid coming up right through the center tube, and so it starts getting plugged up there. So I don't know if I would say the tree makes an effort, but that's just the natural process that it goes through. Yeah.
And then final question, is sapwood different from the cambium layer?
Yeah, the cambium layer is where the actual cell division is going on that grows both the wood on the inner part, which is the xylem portion of the tree, and the bark on the outer part, which is the phloem portion of the tree. So the question was, what was the question again?
Is sapwood different from the cambium layer? Are they the same thing, or are they different?
Yeah, they're different. The cambium layer is the area where the primary cell division is going on, and that's being fed by the flow of nutrients and fluid. But that sapwood is where is like a general conductor of all those materials, whereas the cambium layer is where the cell division itself is actually going on.
So bark, cambium, sapwood, heartwood.
Yeah.
Okay.
Good.
All right. Thanks, Chuck. I'll go on.
Good. Okay. So the characteristics of plantation chestnuts that we're growing in our hybrid plantations there. The specimens that we're getting in the lab here are typically from culled trees, right? And so because we don't mind going in and culling out trees and then using those for specimens, and those typically have a significant impact from the blight cankers, and so that has an impact on the curve, direction, and things like that of our growth rings there. These are chestnuts that are typically in the first 10 to 12 years or less. They're almost all juvenile wood. There may be some older trees out there that has some mature wood in them, but not much. And typically, if we've got nice growing trees out in our plantations, we're not wanting to harvest them yet to look at what their mature wood might look like yet. But maybe we'll get to that point one of these days. They're also saplings. They're mostly sapwood with a high moisture content. They're green and they have a thin bark, so they're attractive to insect pests in most of the tree's lifetime up until the time they're harvested, and that results in this annual deformative impact on the wood development there. So that accounts for all this wood variation that we see in our hybrid specimens when we look at them, when we harvest them and look at the wood. And that's opposed to a mature forest chestnut that the lumber used to be harvested from 100 years ago. There were steady growth patterns in the majority of the tree's lifespan. There was, they had typically thick bark. They were resistant to pests. They were mostly mature wood. The juvenile wood was a very small portion of the wood in those big trees. mostly heartwood with a lower moisture content. So that's the difference between the old trees in that first picture versus the young trees that we're looking now for hybrid wood. I wanted to touch base really quickly with the guy who really started off this whole thing, I guess, at least from my knowledge. Gary Carver is a Maryland member of the Chestnut Foundation. And he is also a wood carver, and he noticed as he was working on the wood that there were just the chestnut. He was working on the hybrid cuttings from his plantation and other neighbors' plantations, and he said, "Hey, this just isn't the same as chestnut wood that I'm used to." And so he wrote up a nice white paper which kind of described the problem. Did a great job of describing that, and even included some pictures, which I think made a pretty convincing argument that something was different, right? So here's two pictures of a piece of traditional chestnut wood here, and it looked like that, and that's what he was used to seeing. And he said, "Hey, this is what I've got now, and it looks like it's a different color, it's got different wood grain properties and all that sort of thing." He presented that to the Chestnut Foundation, and I think they kicked it around for a while. And when Sarah asked me if I could contribute anything to that, I said, "Well, I'd be willing to try." And so that's how the whole thing got started. So thank you, Gary. He's not on the call today. I've been talking back and forth to him this morning, and he said that he's just not cut out to sit through a 1-hour Zoom meeting. And I said, "Yeah, Zoom was not made for active people, but bookworms like me, it's fine." It was also funny. I sent out an email to my mailing list for the followers of my Go Wood blog, and one guy, and I said something like, "Hey, if you're bored at lunchtime on Friday, you might want to tune into our Zoom webinar on chestnut wood." And one guy sent back, "You mean if we want to be bored on lunchtime by Friday?" So we'll try not to make it that. So okay, so Gary had proposed this as a potential research project for the foundation. The board of as far as I understand the process, as Sarah explained to me, the board considered it, put out a bid for a proposal. I gave them a proposal, and the nice thing about it was I was able to acquire some equipment to do some work. I upgraded our facility here at our Penn State. We call it the Penn State Xylarium now because we have one of the largest wood collections in the country now here, with over 4,000 species of wood now, and I think something like 15,000 specimens. But we didn't really have modern equipment. Most of our wood tech equipment that I inherited when I did my transition in my career was from the 1960s, and it was all still analog scopes and things like that, not with computer hookups or anything. Analog cameras, where you still had to develop the pictures and things like that. So I was able, with the grant from the Chestnut Foundation, to purchase this equipment, which is a nice computer, Olympus microscope with a camera. Here's the scope here. Let's see, there we go. Scope, camera on top of it, a nice light source to make sure that we always had a constant light source on our specimens here. Here's the computer, the screen. This was a nice. This was actually the computer that the equipment company that sold me equipment was using this for demonstration. So it was already set up and ready to go. And I already had the scale, but this is the basic setup that I used during the study. And I had it set up specifically so that this never changed. All the specimens were examined from the same height of the camera, so that the focal length here from the camera to the specimen was always the same. The lights, these never moved. The setting was always set on exactly the same setting. And so what we were able to do is examine all the different specimens in our study, which was, I think, close to 70 specimens, which represented 70 different trees, by the way, which was, I think, important in our variability assessment here. We were able to examine them all under the same conditions, which is very important when you start looking at color in particular. Yeah, so and I'll mention that since one of the things that I hope to be able to do is have replications of the specimens so that I could get more samples and increase our sample size and potentially the significance of our statistical analysis. But I started realizing as I did that, some of the specimens that I had were very small, and I could only get one sample from each specimen. And the problem then became, well, if I've got a big log, for instance, I could cut more specimens excuse me, out of it and get perhaps 10 specimens out of one particular tree. But then I'd be introducing bias into the experiment because, tree A might have 10 specimens, but tree B would only have 1 specimen. And so I didn't want to wait, I didn't want to have certain trees having a lot more weight in the results than other trees. So I kept it to 1 specimen per tree, excuse me. And, but I was able to accumulate with Gary's help and Sarah's help and a couple of other folks. I think, yeah, Steve Foy here at Penn State helped us out a lot. And trying to think who else gave me specimens here. But a lot of the specimens came right from our collection as well. And we were able to look at about 70 trees overall. I'm going to show you in a minute here on a slide that I wish we'd have had some more, especially Chinese, the Castanea mollissima, but we can do that in the future or not. So the first step then in the study, once I had everything, my methodology standardized and all that kind of thing, was to develop a color index for use in quantifying the color differentials. So here's a picture that I took that was kind of interesting. These were 5 different specimens that Gary gave me. And we see an American chestnut, a B2, a B3, a B4, and then a Chinese chestnut all in a row here. And this is what Gary was seeing. He was saying, hey, look, this Chinese chestnut looks different. It's greener than the American chestnut. And people kind of knew that. And these are all dry specimens, so you can see they still have maintained sort of a greenish color there. But these B2, these hybrids here, to me, I don't know what you see because everybody's eyes are different. But to me, these look more of the reddish gray of the American than they do of the Chinese. And again, I have to emphasize everybody's eyes are different. The light conditions are different, and all of these things impact what we see as color. I had my cataracts removed about 2 years ago, and you won't, if you haven't had that operation, you will not believe the difference in the brightness of colors once you have your cataracts removed there. So yeah, it's definitely an eye thing. Now I took this picture and I said, well, okay, so it looks like there's a color differential. I took the picture and using the filtering system on the camera, I increased the saturation of the colors to make sure that I could see, to really emphasize the peaks of the color gradients there. And sure enough, you see more reddish here, more green, quite a bit of difference there. And so I said, okay, I've got to come up with a sort of an index that helps me determine how to quantify that color differential. So what I did was I took the specimens and started looking at, this is the screen that I actually have in the software. It gives me the RGB, the red, green, blue spectrum of the specimen under the camera. And that's the measurement of how many pixels are at each color along the wavelength spectrum there. And I can either look at the RGB average, or I can isolate the reds, the greens, and the blues to get those numbers. And I've done both of those now in our study. And so, and this is a neat picture. I just threw it in here for this one. Again, we're looking at Chestnut Rays, which you don't normally see because they're so small. This is at 45x. And so we see rays, and we also see a little bit of reflecting of the light there. So that's kind of a neat picture I wanted to include. But again, just to show how the data was processed and collected from the computer. So what the early data told us in this color differentiation step was that chestnut color is differentiable from other species. How did I do that? I found a bunch of different wood specimens in our Penn State xylarium here, tried to look for ones that had distinct color variation. And I got the numbers just on the RGB scale that's reported by the computer, and I lined them up. So they went from 33 to 117, and this is how they lined up. And you can kind of see that it's maybe not so much a color grouping as it is light to dark, right? And so the RGB scale is basically showing us a reflective measure. The brighter the color, the higher the RGB average, and the darker the color, the lower that number is. The problem with that reporting that I found was that my chestnut specimens weren't grouped, right? And they went all the way from 62 from a mature American chestnut to a 96, a juvenile mollissima. So I said, well, okay, so I've got to do something here to improve that algorithm that gives me a more distinctive evaluation of chestnut color, okay? And so I played around with some algorithms, and I came up with this one. and it looked pretty good, because what I had now was a color index which showed my four, I again lined them up by this color index from 0.076 to 1.49, and I had all four of my specimens were within 5 of each other. And so you kind of had a distinct chestnut range here on my color index from 0.94 to 1.16. This one in the middle here is the Russian olive or autumn olive. And so if anybody ever asks you what color autumn olive wood is, you can tell, well, it's sort of chestnut colored, because it's right in there with the chestnuts there. And then these other species fell out. Of course, there are other trees, other woods that will fall in this range as well. But at least I had an index which now grouped a chestnut color, and I could differentiate the chestnut by color from other species. So that was possible. I threw this slide in because it was kind of an interesting thing to kind of confirm some of my numbers for me. I did have 3 specimens of Castanea sativa, the European chestnut, and I didn't really throw them into the study because we were talking about dentata and millissima and its hybrids. But this was interesting because what I did was these were all what I thought were a combination of juvenile wood and mature wood, juvenile wood and mature wood, juvenile wood and mature wood. And so I had these 3 different specimens, and these together were one piece, these were one piece, and these were one piece. So I got the density of those, and I had a 34 pound, 34.2, 33.7, and 31.9. But I said, well, but those are the densities of combined juvenile mature. And in my data sheet, I wanted to have specific numbers for juvenile versus mature. So I went ahead and split those specimens and recalculated the densities and reperformed the microscopy on them. And then I found out that sure enough, we do have different densities. The juvenile wood was 35, the mature wood was 33. The juvenile wood was 34 here, the mature wood was 26. And this was one of the early signs to me that the chestnut was different from other woods, because the juvenile wood was actually showing a higher density than the mature wood. In this particular specimen, though, when I split it, the density was almost exactly the same on both sides of the split. The same thing with the color index. I had a 0.068 and 0.054, a 0.64 versus 0.58, showing a color differential between 2, but again, almost no color differential at all. And so I went back and I said, what made me think that this was juvenile wood? And it just so happened that the way that the specimen looked, with the whiter rings and the fact that it wasn't sanded and that sort of thing. And then I realized, well, this is probably not juvenile wood. This is mature wood that just looks a little bit different from this mature wood, but it's essentially the same. And so I was able to go back and reclassify this specimen as a mature wood specimen. And it also confirmed for me that my findings and my metrics confirmed for me that my density calculations were doing well, and my color index was also doing a good job of differentiating between juvenile wood and mature wood. So a nice confirmation of my numbers there. So yeah, sure.
Sorry to just because I have a question about what are the density units. So just to clarify, what are you?
Oh, I'm sorry. Yeah,
with density?
That's pounds per cubic foot, right?
Okay.
So you can imagine a cubic foot of wood. That's how many pounds those numbers represent. 35 pounds per cubic foot. So okay. Now, the next thing that this early data told us was that the chestnut color, while chestnut was differentiable from other species, it was also differentiable based on juvenile or mature wood. And so this was a good example. We had a millissima juvenile, which had a color index of 4 versus dentata mature, a 0.90. And so you can see that they're different color, and the color index was picking that up. So that's juvenile mature. Oops, I thought I had more examples of that. But we saw that pretty consistently throughout the specimens, that the juveniles were on one side of the color index and the matures were on the other side of the color index. So that was good. The other thing it told us, though, was that the chestnut color was not differentiable by species within juvenile and mature wood classification. So in other words, once you were within the juvenile classification, you couldn't tell the dentata from the millissima. And once you're in the mature wood, you could not tell dentata from millissima. And this was a good example. This is juvenile wood. These are both juvenile wood. This is a B3F2 specimen that was freshly cut, 6 weeks, and had 6 weeks of drying in my lab, 4 rings per inch. The color index was 0.80. Here's a 100-year-old dentata, 100 years old, juvenile wood, 10 rings per inch. The color index was 0.82. So both juvenile wood, you cannot tell the difference between the dentata and the hybrids. And I would assume that that would also, it might be different for the Chinese, but I don't, my recollection is that it's not. This is a sort of a confirmation of that. This is a radar plot showing the RGB averages for the 3 different species, American, Chinese, and European chestnut. And you can't see 3 different triangles, but they're there. Which shows basically that the chestnut color is one particular color pattern with respect to RGB averages, and it's almost exactly the same, regardless of which of the specimens that you're looking at, species you're looking at. And this would be a reflection of all of our specimens, and that's juvenile and mature wood combined. Okay, so a chestnut color really is a chestnut color. Now, the interesting thing was I put in our hybrids, and the hybrids then had the same color shape, but they have higher peak averages on each of the RGB scales, which simply means that they're getting more reflectance from the same amount of light, which means that they're a lighter color. It's the same color, but it's a lighter shade of it. Okay. And so that's a reflection of the fact that the hybrids are just juvenile wood as opposed to these other 3 triangles, which were a combination of juvenile and mature. Okay. And then just for fun, I threw in the juvenile wood from American. And again, we saw a very similar pattern. It happens that it's a little bit darker because it has a little more reflectance on the blue scale, but basically it's the same shape. And interestingly, it differentiates itself from the averages of the patterns, and it goes more towards the hybrid juvenile specimens there. So that's an interesting thing that kind of gave me an idea that we're on the right track here by comparing juvenile to juvenile, etc. So then I had to, but then at some point I felt like I didn't have enough differentiation in my color index. And so this was my original color index that I was using for that stuff that I've been showing you. And so this is a pairing of mature versus juvenile Castanea dentata, the American chestnut. And so these are, I think, 17 specimens versus 11. And the pairings don't mean anything except that I took the same data with the same pairings and ran them under a new algorithm that I had developed. And I did, in fact, find that I had more differentiation here. This pairing right here actually got a little bit closer, but from then on, I think we have a differentiation of 0.06 versus 0.08. These are about the same, but you can see these 2 are farther apart than they were before. These 2 are farther apart than they were before. These 2 are about the same. Again, these are farther apart, farther apart, farther apart, farther apart, farther apart. So I found more differentiation. I was able to do a better job of differentiating the colors within the chestnut color itself. And I felt pretty good about that. So I used that for the rest. When I went back and started the study over on the final specimens, I stuck with this one color algorithm. Although I calculated the metric for both algorithms just to be able to have it and compare it. Now, these are 2 green Chinese specimens, right? When I say they were green, they were dry, obviously, but they were green from a color spectrum standpoint. These were the 2 specimens in the sample of the Melissima that had the highest green peaks, 122 and 141, and this one was significantly greener than this one. That's a pretty big difference there in green, but the next green was about 100. So these 2 were the kind of like the 2 greenest specimens. And we can see here that the interesting thing here is if you look at the profiles of the 2 specimens, you can see that this one goes like this, whereas this one is much more balanced between the red, green, and blue. This one has a higher blue, and that's creating a reflection of the darker color here from this late wood growth ring. This tangential view is showing us an early wood and late wood but it's not as dark as this part of Lakewood because this tangential view would have been like cut right across here instead of up here or so. So we see two different, quite different numbers on our RGB scales, quite different on our our color index 2 and our color index 3. This is keep losing that, 0.59 versus 0.68. And in 3, we saw that I got increased differentiation. That's a 0.09 differential. This one, it went from 0.24 to 0.39. So now I'm up to a 0.15 differential. So I, in fact, did increase my discrimination there on those colors with my new algorithm. Okay. So that's 2 green Chinese specimens. Now, these are 2 American specimens with distinct color values. One of them is a transverse and one is a tangential, as in the 2 previous pairing. And again, we have 2 significantly different color profiles, 5.8 to 6.3 and 0.2 to 0.34. 2 different colors. You can see it on the screen, 2 different colors. Look at the profiles, 2 distinctly different profiles on the RGB scale. So I thought, well, that's neat. Now let me then switch partners to make a comparison. So now I put the 2 transverse views together. The one's a Chinese juvenile wood transverse, the other one's American juvenile wood transverse. And now all of a sudden, look at these profiles. Pretty much look very similar. Look at the numbers. 120 RGB average to 122. 203 red to 203 red. 122 green to 123 green. 34 blue to 39 blue. The color indices, 63 to 68 and 34 to 39. So very similar color metrics for these 2 transverse view juvenile wood specimens. One's a Chinese, one's an American. Okay. Now we've got a Chinese and American juvenile wood specimens, but now we're looking at the tangential views. And again, look at the color profiles, very similar. The RGB averages are 137 to 142. The red 222 to 228. Greens 141 to 145. Blue 48, 40. Again, very similar, very slight color variation if you want to be that discriminating. The color index picked that up, 0.20 to 0.24. And this is an example where you can clearly see the increased discrimination. The original algorithm that I was using had this as a 0.58 and this one as a 0.59. And I broadened that here to a 0.20 to a 0.24. Okay. So again, we kind of see how now I feel pretty good about our color discrimination because not only if I back up here, these look different colors, these look different colors, and the color indices tell us that. And then when we start putting similar colors together, the indices tell us that as well. So we're looking pretty good. I'm feeling good about going forward and compiling the data and making some graphs. So I do that. And we start looking first at the color index calculations. This is the color index number 3, and I just grouped them all together by mature wood and juvenile wood. So the American mature wood color indexes, the box and whisker plot for those look like this, versus the juvenile wood of the American is down here. So you can see that the color index numbers are the inverse of the RGB reflectivity scale. On the RGB scale, the higher the number is, the brighter the higher the reflectance, the lighter the specimen. On these indexes that I've calculated, the lower the number is, the brighter the specimen is. And so we see that here, the juvenile wood is brighter than the mature wood. And we see the Chinese color, and this is interesting. If we look at the mature wood, the Chinese color is really just a subset of the American color. There's really no difference in the mature wood. In the juvenile wood, perhaps a little bit of a difference there, perhaps a little bit lighter color, and that might be a reflection of that green that we see, which is less pronounced once the wood is dry, right? But the interesting thing is now we throw the hybrid in here, and the hybrid, which is all juvenile wood on our color index, is pretty distinct. It's certainly distinct from the American, completely distinct from the American mature wood. So when Gary was sitting there in his shop comparing his American mature wood, that from his lumber, old barn wood and things like that, to these hybrid specimens from the plantations, he was exactly right. He was seeing a different color wood for sure, right? And it's even different color from the juvenile wood of the two species there. But that wasn't really enough of a breakdown for me to feel comfortable about anything or say anything until I'm sorry, back up. So yeah, so we had a hint there that our color indexes were good. We definitely were seeing a different color from the hybrids and the mature American wood. And so that said, okay, but what is that attributable to? And the first thing that occurs to us is, well, we're talking about different densities of wood. So the next step was to investigate the role of wood density in the color variation.
Hey, Chuck, before you
Yes.
go on, I've got a lot of questions about the color index.
The what?
The color
Okay.
index. So
Okay.
someone says, how do you determine the color spectrum? And then I also have a question about color index. Steve asks, what is the scale being used? Is it relative to a base white balance? If it is an average of RGB values, and it is more of an indication of luminance or gray value. I think that there's some confusion as to what exactly the scale is.
Yeah.
I don't know if you can sum that up.
It's relative to it's the differential color of the peak values of each of the RG and Bs and relative to the RGB average. Okay. And so what I was trying to do was separate out the particular effects of the influential red, green, or blues. The blues were not so influential because they tended to stay right around the same numbers there for all the specimens, but there was more differential in the R's and the G's. And so I played with the ratios of those, and then to keep them all on the same scale, I normalized them by the RGB values. So I think that the person that asked that question will understand that answer. The other one had to do with what was the first question, Sarah?
I think just how the scale was determined, but I think.
Oh yeah, no, okay. The scale itself, though, comes from the calculations that the computer makes. What it does is it goes across the color spectrum and determines how many. It takes that picture and it determines how many pixels of each color are in. How many pixels there are of each of the 256 colors across, and that's what causes those curves there. Okay, so they're population curves based on how many pixels there are of each one of those colors. Okay. All right, so that's color. Now we investigate the role of wood density in the color variation. So I started looking at the wood density of the specimens, and this is the American chestnut, juvenile versus mature wood. What we found here was that the average density of the chestnut in our sample was 32 pounds per cubic foot. The juvenile wood was 36 pounds per cubic foot, and the mature wood was 30 pounds per cubic foot. So this is one of those things that said, well, this goes counter to what most publications and most books and papers on juvenile wood kind of say the opposite, that juvenile wood is lighter than mature wood. But there's a reason that I'll talk about towards the end of the presentation of why I think this is going to be pretty consistent across chestnut. We did have the juvenile wood exhibited 5 rings per inch on average. The mature wood was 12 rings per inch. And this is the RGB average. We see a higher reflectance value of the juvenile wood versus the mature wood there. So the mature wood's a deeper color, darker color. All right, so that's American wood, 32 pounds average, 36 juvenile, 30 mature. If I then go to Millissima, the Chinese, we've got a much higher 45-pound average, about 9 pounds per cubic foot higher, 44 for the juvenile. There's less differential between the juvenile and mature. And that would agree with like a paper that I talked about. I think I mentioned this Black Locust paper, where they found no significant difference between the juvenile and mature. But you would think that you'd see a lighter density on pounds per cubic foot. This number I'm not feeling good about because I got this very small sample of only 3 mature Chinese specimens in the study. So I'd like to increase that number, and I'll be talking to my IWCS, International Wood Collectors compatriots, to see if I can get a few more into the study here and see if that changes that number at all. It might not, But we do see that we had only 8 rings per inch on our mature Chinese specimens. If I had a lumber from a bigger tree, we'd have more rings per inch, and that might change this number somewhat. So we'll see how that goes. But again, the RGB scales were about the same as the American. So that's telling me that, okay, that's what the data was, but what's the specific effect of density on color? So here I've plotted scattered plots of the American mature wood specimens versus the American juvenile specimens. And here's the mature wood and here's the juvenile wood. And you can see that they kind of occupy at some point the same space, but also maybe a little bit different space. The juvenile wood tends to be a little bit higher density, as we've already said, whereas the mature wood, which is all of this plus this, is sort of lower density and a higher color index, a darker color. Okay. Now the interesting thing was I plotted the regression lines through these small set of data here, and we see that there's really no, because of the horizontal flatness of these regression lines, we see there's really no significant response relationship between color index and density. And I think that's going to hold true through the chestnut colors, although I'm going to show you something a little bit different here in a minute, but this would not apply between species. So if we go between species, if you think back to the first slide I showed you where you had all the different species of different colors, there probably is a definite relationship between density and color. Okay, the darker, the heaviest woods in the world tend to be a darker color, and the lightest woods in the world tend to be a lighter color. But in this intermediate range here, probably not as much of a relationship. Certainly within a species, probably not a relationship at all. Then I plotted the Chinese mature and the Chinese juvenile, and you can see we're occupying a different space. It's a higher density and a bit lower on the color index scale here. So I only put this here to introduce the next slide, which I think is really one of the more interesting slides of the presentation. And that is when I introduce the hybrids into the graph, I see an interesting conclusion that I think your cross breeders out there will love this slide, because what's basically saying is that, okay, here's my American, here's my Chinese, and look at the hybrids are right there between them, sort of in that transitionary zone. And the fact is that those are all juveniles. So they are kind of in this direction from the Chinese juveniles, moving sort of more in line with these American juveniles. We do show, when I plotted these regression lines, we do see a relationship between density and color, but this is in the juvenile specimens here. And once you combine the juvenile mature, I think you're going to wind up with essentially no relationship there. If you think about a correlation line going through the middle of all of this data, it's going to be pretty flat. Okay. Now, to make it even more interesting, I split the hybrids out by B2s, B3s, and B4s. And we see here, and now we only have juvenile American and juvenile Chinese on the graph. So here's your juvenile Chinese, here's your juvenile American, the blue, and here these multiple colors are the hybrids. We see that the B2 started out on the higher color index side and a little bit higher density. So they're kind of more like the Chinese in some ways, but leaning towards the American. And then you got to the B3 and B4s. And if you want to say the B3s and B4s are all the same cluster, you can say that they're kind of indistinguishable from each other. And they are moving towards the lower density and lower color of the American, lower color index of the American, again, in that transitionary phase between the Chinese and the American. And if you really want to stretch it and be optimistic, you could say that, look, the B4s are kind of moving even more into this American direction. But that's a very small sample size. It's 4 pieces of data. I wouldn't draw that conclusion at all. I would probably tend to say that this is all the same subset, and I'll say that in our conclusion to the study. Now, let's look at another interesting relationship. Here's those B2s, B3s, and B4s again in relation to American mature wood. and they lay like this. Here's the American mature wood. And that might remind you of something we've already seen. That might remind you of this, which is the American mature versus the American juvenile. The juvenile, again, sitting in this direction, the southeast direction from the American mature. And that's the same relationship we see here with our hybrid specimens as well. So we're feeling pretty good about, at least based on this measure, the color index versus density correlation, we're feeling like that the hybrid specimens are kind of positioned where they should be if they're going to turn into a mature wood that resembles the American chestnut. So I asked the question, if density is not the principal reason for the entire specimen color variation, what causes this color variation? Are the cells themselves a different color, or are the cell patterns that cause the difference? That leads us into this next area of investigation. Step 3, investigate the role of cell type, size, and variability in the color variation. So we start looking at these cells with the scope, and we start taking measurements of the relations of the size of the growth rings, the size of the early wood, the late wood, and the pores themselves, the first ring of pores. And we do this. This is an interesting picture of it's a transverse view that we're looking at, and I'm showing how I make the measurements, and I record this data. And this one I just threw in here because it has the edge of the wood here, and it's showing sort of it's not exactly a tangential or a radial cut. It's sort of like a quarter sawn cut, and you kind of can see the quarter sawn look here at the edge of the wood here. So that's kind of interesting. But here's a transverse view of a mature wood with 4 growth rings, 4 really nice size growth rings. And we can clearly see the width of the pores themselves, the total width of the early wood, which includes the pores, and then the width of the mature. I'm sorry, I'm getting confused on the terminology, as a lot of people do. The total width of the early wood, which includes the pore wood and the later early wood, and then the total length of the late wood, and I don't necessarily go completely by color. If I looked at these hundreds and hundreds of times, what I started sensing was that you can tell, again, we go back to these fibril angles and things like that. You started seeing these angles, these fiber angles start to get more uniform and straight in the mature wood. And that's how I determined where to bring that line, partly with color and partly with the uniformity of the parenchyma alignment and things like that, okay? So, I took the measurements on all the specimens in the study. I recorded the RGB. I took these measurements and recorded them. And some of the views were transverse or cross-sectional, like that one. A few, about a much smaller number, were radial views like this, where we're looking again along the growth rings, but now we're looking down the side of them instead of across the top of them. And we can see the depth of the pores themselves. Now, this was an interesting picture. I said, well, do I get the same results if I measure the width of the growth rings in this radial view as I do from the tangential view? And sorry, before I get into that, this was an interesting thing I threw in about tyloses. If you know the story about tyloses, and I assume almost everybody's heard this story, but I'll repeat it very quickly. And that is that tyloses are occlusions that clog up these pores. And the main difference between red oak wood and white oak wood is that the white oak wood has tyloses clogging these pores, and the red oak wood doesn't. And that's why white oak is used for barrels, because they don't leak, and red oak can't be used for barrels because they do leak through those pores. So chestnut has tyloses as well, and a pretty high percentage of tyloses in its wood. And this was an interesting quote that I got from a fairly recent paper here. She says, she gives a good summary of why tyloses are there and what they do. She says, vessel occlusion through tyloses or gums is a natural phenomenon occurring with aging and heartwood formation, and in sapwood in response to vessel embolism. These types of vessel occlusion play a crucial role to limit the spread of pathogens and wood decay organisms, also as a part of compartmentalization after wounding. In the sapwood, they can be considered to be an effective stress response. So does it make sense that chestnut specimens would have tyloses in, considering all the stress that they're under and everything else? Yes, and all the stress they've been under for the last 100 years, the answer is yes, it does make sense that they would have those. So just an aside there about tyloses, okay? Now, I went in with that picture from before and used the computer to measure, to define polygons around what I could identify as poor wood versus the more solid, more solid wood in the growth ring. So I identified those polygons, measured them relative to the entire sample, and saw that it took up 44% of the area. And I said, okay, so hopefully that will confirm my other way of measuring. And so when I went back and looked at the same piece and looked at my transverse measurements, averaged them out as I did here, and came up with my pore to growth ring ratio, which is what we saw in the previous slide. I came up with a 0.4 something. It's rounded to one number, unfortunately. But yeah, I got the same number from a different view, which made me feel, again, real good about my methodology there. This one was interesting. This is a tangential view. This is one I alluded to earlier here. We're looking at a flat sawn piece of wood. So you're looking at the top of one growth ring here. And you're seeing growth, you're seeing rays coming through here. So in this picture, imagine that the pith of the tree is right here in the middle of the piece of wood, but it's behind the piece of wood, behind the screen. And the rays are emanating out from the pith. And so here in the center of the picture, we see these real narrow bands of the rays, and that's sort of the actual width of them. All right, whoops, back up. That's the actual width of them. Now, as we move farther to the sides, what we see is now you can imagine that instead of these rays coming directly at us from the center, now what they're doing is they're emanating outwards in other directions because they're shooting out in a radial direction. And as they go out at different angles, now when we saw flat, these rays start getting wider and wider as you go. So that's a neat way of thinking about how the ray width variation occurs in especially when we see it in oak. You don't see it so much in chestnut because these rays are very, very small, and they don't show up as clear as this. This is 7 times real size here. So, okay. And so I was able to take a radial view and measure them the same way as I measured the transverse views. Again, I don't think I mentioned, I tried to take 3 growth rings from each specimen and average those measurements to use in the study. Again, I've got 3 growth rings here on a tangential view. Some of the tangential views, the growth ring was too wide, like in that previous one where I showed you the rays, so I didn't get any data off of those. But in this one, I was able to see 3 growth rings on the tangential view. I use these, and it is interesting that I found that the relationship in all 3 views kind of held up. In fact, it did hold up. I saw similar ratios, whether I recorded those in the tangential or in the radial or in the transverse view. So that's a good thing. So now we get to the results of the study, of the final phase of the study. And that is, we're looking at the growth rings. Nothing really significant here in just the width of the growth rings. We're seeing that generally speaking, the mature has narrower growth rings in the Chinese and the American, and that the hybrid has wider growth rings. And they seem to be a little bit different from the juvenile wood of the Chinese, but really there's not a lot of significance going on there in this slide at all. The growth ring size is more a function, I think, here. The total growth ring size is more of a function of the growing conditions of the tree. Now, though, we start looking at the ratios between the components of the growth rings. And I broke them up into those 3 components: the pores, the total early wood, and then the late wood. And here I started looking at the relationships between them. When I looked at the pore to early wood ratio, I found that we see a difference here in the American between the juvenile wood and the mature wood. But the more significant finding here is that the hybrid pores more closely resemble the Chinese pores than they do the American pores. And that's almost a significant difference there, almost distinct there, where these are definitely a subset of the Chinese. So in this case, the pore to earlier wood ratio of the hybrids is more closely closer to the Chinese than the American. The same thing when I get into poor late wood ratio. Sarah, did you have something?
Yeah, we're
Yeah.
getting close to the end here, and I
Okay.
guess what I did want to mention though with the hybrid is, maybe in a write up it would make sense to break apart the B1, the B2s, B3s, and B4s, because I think it's hard to lump all of them in and say, well, hybrids are closer to Chinese, because I think what you saw when you did break them up is that as you back crossed, the earlier back crosses acted more like Chinese, but then the later back crosses acted more like American. And I think, being able to break them out, rather than saying, well, all these hybrids are acting more like Chinese, I think that might make more sense. And just to make that clarification for the listeners. But we've only got 4 minutes left, Chuck.
And I'm almost done. Yeah.
Okay, there are tons of questions. So what I'm going to do, I'm going to let you finish.
Yeah.
But I'm going to let everybody know, we're going to probably stop at 1:00. If you have a remaining question in the Q&A, Chuck, I hope that you wouldn't mind answering some of those questions via email.
Yeah, sure.
And
absolutely.
I can send those to you. And then I do have a record of everybody. If you gave me your real email address, I'll send this back out to you. If you didn't, then send me another email and we'll get it answered for you. But there's Kendra and I have tried our best. We are not wood experts. So we've tried to answer some of these questions, but we weren't able to. So we'll follow up with those if you have some remaining stuff. Thank you, everybody. The chat was fantastic. Chuck, we'll wrap up and then I'll give a final closing for everybody. Thanks.
Okay, I'm gonna just hit these slides about 10 seconds a piece here, because we really got to get to the final slide to kind of get to what you're asking for. I look at the pore to growth ring ratio. Again, the hybrids are kind of transitionary between the Chinese and the American, but very, very distinct from the American. But once we get away from the pore ring itself, the early wood to growth ring ratio, we see the hybrids are much more like the American juvenile than they are the Chinese. When we get to the late wood to growth ring ratio, again, the hybrids are very much more like the American than they are the Chinese. And the most important one I think of all is the early wood to late wood ratio. And again, you see the hybrids seem to be a subset of the American juveniles and very distinct from the Chinese. So these are all really good indicators. And I summarized them by saying the hybrid growth rings tend to be wider than either the American or Chinese growth rings. The hybrid pore width relative to early wood is more like the Chinese juvenile wood. But in the three ratios not related to pore width, which are the early wood to growth ring, the late wood to growth ring, and the early wood to late wood, the hybrids almost precisely match the American ratios and are distinctly different from the Chinese ratios. So the final conclusions, in color, RGB averages show no difference between the three species, but the hybrid specimens are lighter. The color index examinations definitely show a color distinction between American wood and Chinese wood. Chinese juvenile wood, but not mature wood. And the color index examination also confirms that all hybrid juvenile wood specimens are distinct from American wood. So that explains the color differential that Gary was first concerned about. In density, the Chinese wood is more dense than the American wood. The hybrid specimens were 10% more dense than the American specimens, but were almost precisely the same density as the American juvenile wood. So the density profile of hybrid chestnuts is much more similar to American chestnut than the Chinese chestnut. The correlation plot suggests that within the chestnut color, there's no correlation between color and density, but those same correlation plots do show that the hybrid specimens occupy the same color density profile as American juvenile wood and have a distinctly different color density profile than the Chinese chestnut wood. And in the cell growth metrics, the American chestnut, a mature wood, goes against convention and is less dense than its juvenile wood. The hybrid specimens exhibit the same approximate growth ring size as American and Chinese juvenile wood and distinctly different from the mature wood specimens. The hybrid pores exhibit similar characteristics to Chinese wood, and the pores exhibit similar characteristics to Chinese wood, but in all three ratios not related to the pore ring width, which are early wood to growth ring, late wood to growth ring, and early wood to late wood, the hybrids almost precisely match the American ratios and are distinctly different from the Chinese ratios. And so the implications of the research, and this is the last slide, I think. The color variability in the data suggests that the hybrid wood is more nearly Chinese colored, but this is due to the color metrics only looking at juvenile hybrid wood. The stand parameters of hybrid plantations mirror more closely Chinese chestnut stands in growth form, promoting wider crowns to stem and more fluid in wood transportation during early wood generation, which results in a pore ring and early wood size that's similar to the Chinese trees. However, the annual ring data suggests that as hybrids evolve from juvenile wood to mature wood and crowns close and thin, the wood will more closely resemble American chestnut wood and will be distinctly different from the Chinese. The final form of the tree will determine the ultimate wood quality. The taller and more tubular the tree, the more the wood will resemble the old American dentata. And finally, the color density profile suggests that the F3 generation of hybrids, in fact, moved wood parameters closer and perhaps indistinguishable from native from Castanea dentata, and were a significant improvement over the F2 wood characteristics. We've got a small sample size on the F2 and F4, but you can kind of see the trend there. The small number of F4s in the study seem to suggest a slight improvement over the F3, but so slight as to probably not be noticeable or statistically significant. Okay, so we are seeing the trend, but possibly the F3 in terms of wood quality is as good as it needs to be. And that's the end. We can sit back and enjoy our chestnuts roasting by the open fire there. And I guess there's no time left. I'm sorry for running over here, folks. I had no real idea I was going to talk that much about some of those slides. So
well, I know what to do with some early questions too. Usually we don't end up doing that. Well, we did end up having over 200 people. I think a lot of people had to take off a little bit early. But Chuck, thank you. This was really fantastic. There was so much really good chat in there. The Q&As are still there, so there's a lot of stuff to follow up on. We may even need another chestnut wood talk about some other aspects. So Chuck,
well, I just want to thank, like I said, this is by far the largest audience I've ever had for any kind of wood science. I thank you so much to the folks for tuning in and spending time with us today. And I will be glad. My email address is cdr Charles David Ray 14 at psu.edu. So if you want to send me questions, I'll be glad to answer them all.
Great. Thanks so much, Chuck. And I'm sure I'll see you around campus. And I hope everybody has a great weekend. We will be back in October, the third Friday in October. We're going to be joined by Nathan Cood and Jared Westbrook. And they are going to be talking about genome sequencing. And we're going to take a tour through the newly published American chestnut genome that was published to the internet, I think in the summer. So I hope you guys can show up for that too. That's going to be a great thing. Learn what genome sequencing is, how it's done, and how we apply that information as well. So thanks everybody for joining us month after month, and I'll look forward to seeing you guys soon. And go Penn State. Hopefully we win on Saturday.
Yeah, really.
All
Thanks,
right.
Aaron.
See ya. Bye.
Bye.